Thank you for your kind presentation, and thank you to the organizers for giving me the opportunity for being here. Again, we are going, or we are getting normal, so we can meet, we can have symposiums, which is always nice. So, and well, the first slide is just to show you the group, the people who does the work. As you can see, we are an open arms laboratory, so we have received people from 26 different countries. And as you can see, we are continuously working a lot. So this is, well, no, it's really the barbecue of every year, okay? And this is the best part, of course, in the laboratory, because you know the people working in the laboratory, well, some of you still are working in the laboratory, you know how is the daily work, okay? It's more or less like this, so it's not that fun. And many times when you say people, well, I'm working in science, they think, well, this is really nice, so you can be every day saying Eureka. Well, no, it's not true. More or less, the life goes like this in the lab, okay? And in a lab with a lot of instruments, the every day is something like this, okay? So we are suffering a lot. Of course, an important part is when you want to make the knowledge just public, so you need to publish your work and you send your papers. And then many times you are able to find this kind of reviewers. This is the reviewer too. And this is what he did with your paper. So yeah, life is complicated in the laboratory. But anyway, I just want to take this opportunity to invite you, if you want, to send a review paper to this journal. It's a nice journal. The impact factor, I think, has increased this year a little bit. And also you can send to this other journal. The impact factor, I think, has decreased this year. So, but they are, your papers are very welcome. Okay, so let's go to business. And I would like just to give you a few ideas that probably all of you already know, but just in case, about the situation of food science, the future of food science, the challenges that still we have to solve, okay? And as you can see, this is some of the really crucial points. So you know that paradoxically, as billions suffer lack of food, more than 2 billion people, 30% of the world's population are overweight or obese. And we will need to close a significant food gap by 2050. So due to the population growth, that you know it will be a problem, and of course the changing diets. So more than 1/3 of all food produced globally goes to waste. This is really a critical point too. Regarding food safety, so we are continuously working on that, but the World Health Organization estimates 600 million, almost 1 of 10 people in the world, cases of foodborne illness, and 420,000 deaths every year, 5,000 in Europe, okay? The agri-food production, including manufacture, food preparation, and cooking, accounts for approximately 30% of all the greenhouse gas emissions, and livestock production accounts for approximately 50% of this. But this is also the point that we have this link between foods and non-communicable diseases, and we need to change this for nutritional food safety. So we have the problem of emerging foodborne pathogens that are changing the epidemiology of foodborne diseases. Some chemicals, of course, have of concern too, and food is a major source of human exposure. But look at this. We have seen this morning, David told us a lot about these food components, but only 150 nutritional components are tracked in food composition tables, okay, in the labels of the foods. So about 0.5% of the 26,000 approximately chemical compounds documented in food. This provides a very poor representation of this complex issue. And of course, we need climate smart and environmentally sustainable and safe food systems linking biorefinery and bioeconomy. And this is the concept. So just to make a better use of what we have around, okay? And of course, on the other side, the 21st century is also observing a technological revolution that includes breakthroughs in nutrition, genetics, omics, informatics and bioinformatics, global networking, remote sensing, precision farming, low-impact agricultural, sustainability, biorefinery, and One Health concepts. These advances will hopefully continue to drive major global changes in our daily lives. To get so, it is of vital importance that countries and governments around the world recognize the opportunities and challenges and provide the appropriate framework support, investment, and infrastructure. This matter of politics, you know it, so be ready, because this is Bertrand Russell sentences. Scientists try to make the impossible possible, politicians to make the possible impossible. You know it. But also this is a good one, that I like it. The fundamental cause of the trouble is that in the modern world, the stupid are cocksure, while the intelligent are full of doubt. And well, we know many times these politicians in which area are. Okay, so this is also some of the challenges that we have to face. So look at this, because we have in front a lot of work to do. So we need to reduce through diet the impact of cardiovascular diseases, obesity, cancer, and why not Alzheimer, to understand the effects of gene-food interaction on human health, to explain the different answers from individuals to food, to establish the global role and functions of gut microbiome, to understand the stress adaptation responses of foodborne pathogens, to understand the molecular basis of biological processes essential for improving agronomic and farm animal production, to understand post-harvest phenomena through a global approach, to reduce food allergy, to carry out pangenomics, of course, to improve all the bioinformatics, and to get a personalized nutrition. How far we are? Well, in my opinion, we are just starting to walk this very, very long way to get this personalized nutrition. And of course, it's far, far away still. So we have there a lot of work to do. So that's why a few years ago, we defined this new discipline, Foodomics, as a discipline that studies the food and nutrition domains through the application and integration of advanced omics technologies to improve consumers' well-being, health, and knowledge. Basically, what we try to do here is to use these massive techniques of analysis, metabolomics, proteomics, transcriptomics, and to face topics regarding bioactivity, but also safety, quality, and traceability of foods. We have done for years, we have worked a lot of colon cancer, and basically our idea there was not to cure colon cancer through the diet, but just to decrease the evolution of the illness, okay? And well, we did a lot of work. If you are interested, you can find many of the papers that we have published on this topic. But what I want to talk to you today is about our new work regarding Alzheimer. So practically nothing of this has been published yet. So we have been working a lot on this issue. And why Alzheimer? Well, you know, we are getting older and older. The life expectancy is increasing. We have a low mortality rate. So the socioeconomic development, better health care, of course, but we want to get a healthy aging. The problem is that we have this problem of dementia, of which the main cause is Alzheimer disease. And you know, Alzheimer evolves with the formation of these beta-amyloid plaques of these neofibrillary tangles, okay? Basically, the problem of Alzheimer is that it's a multifactorial illness. So it goes with oxidation, reactive oxygen species or reactive nitrogen species, oxidative stress, microbial process, cytotoxicity, mitochondrial damage, degeneration of synapses, this glucose decrease, this neuronal death, etc. Okay, and as you know, the problem is that there is not cure for Alzheimer. So we are really at the beginning of the illness, even when it was discovered 100 years ago. Of course, the objective in our case was to study the activity against Alzheimer of bioactive compounds from natural sources and food byproducts, following a multi-target approach. Since the illness is multifactorial, we wanted to approach it in this multi-target way. And for that, we have made the chemical characterization by metabolomics and for confirmation of this bioactivity using foodomics with a special emphasis on lipidomics, as I will show you. And we were extremely happy when a few weeks ago, in April, this paper came out from Nature, in which they demonstrate how by the formation of the beta-amyloid plaque, the cell, the neuron, starts generating a lot of ROS, reactive oxygen species, that finally are capable to destroy the DNA and to originate the death of the neuron, okay? So then it's clear that this activity against oxidation, against this species reactive of oxygen, etc., would be a good target, and that's what we did. We have selected several food byproducts. This citrus, this from the orange juice production, this curuba, the seeds of this, this is a tropical fruit, this uchuva, the calyx, the outside part of the fruit, olive, that is very important in Spain, of course, as you know, the leaves of the olive oil production, and the silver skin of the coffee production. But also we have included some plants as Kalanchoe, Rosmarinus, or some other products of the winemaking production, these copperacea buds, and also different microalgae that we wanted to study this possible bioactivity against Alzheimer, okay? We selected green extraction techniques to be able to obtain green extracts capable to be used in any food activity, okay? And with these green extraction techniques, we combined different in vitro assays, one of them just to investigate the activity, antioxidant activity, and the activity against the reactive species of oxygen and nitrogen. This is for anti-inflammatory activity with this LOX. It's an enzyme. This LOX is related to the cognitive impairment, as you can see. Also, we wanted to investigate the anticholinesterase activity of the struts. So this enzyme, acetylcholinesterase, increases in the cases of Alzheimer and destroy the neurotransmitter, the acetylcholine, and then people is losing also the synapses. Also, this activity against this acetylcholinesterase, of course, we also model the blood-brain barrier, okay, that is necessary to be crossed, and this just to demonstrate how some compounds are capable to pass the BBB, some of them are not. And of course, we wanted to characterize these struts and this activity by using metabolomics. In this case, we combined gas chromatography and liquid chromatography with high resolution MS. And this is more or less all the workflow that we follow, just combining the natural product, how it was extracted, how it goes to the analysis by ILSI, how can we go to the databases and to get some chemical characterization also using the MSMS fragmentation pattern. In the case of GC, we were using mostly the NIST library. This together with in vitro reactivity as testing, so we can make a chemical characterization and tentative identification, or even with a standard just to confirm the identity, okay? This is what we did, for instance, in the case of the passiflora. This is the characterization by ILSI and GC, okay? So you see different compounds there. In the case of this is Pisalis, the Calis, okay? These are polyphenolic, butanoids, sucrosa esters. These are, for instance, the compounds, the terpenoids in the strat from olive leaves. These are from Kalanchoe, or this from rosemary. Rosemary, we have worked a lot with this plant. And these are from microalgae that are mostly carotenoids. The compounds that we have found, this is Indunaliella, this is Hematococcus, most of them were astaxanthin compounds. This is the cooperative woods that were mostly these polyphenols. And this is just to show you a comparison of the activity against acetylcholinesterase of the different strats. And this is the drug that they give to the people suffering Alzheimer. As you can see, some of them are really active, okay? This is butyrylcholinesterase, that is also another enzyme that goes up. This is the same again, the enzyme there, the drug, sorry, some of them were even better. This is the anti-inflammatory. This is the standard quercetin that is used. Some of them, many of them were very good as anti-inflammatory. This is the antioxidant. This is ascorbic acid, the standard, or this is Trolox. You see many of them were also much better. And this is the ROS, the reactive species of oxygen. So many of them, this the standard, some were very good. Also, this is against nitrogen species. This is ascorbic acid. And this is just to show you that we select from here the 5 that we consider, it does not go off. No, okay, 5 here were selected as the best ones. And next, what we ran was a study about the toxicology of these strats. For this, we were using human kidney cells, these human monocytes, or these human neuroblastomes. And as you can see, in the case of Dunaliella, at this concentration, we observed some toxicity, so it was removed, this concentration for the study. The gain for monocytes, the 20, was observed to give some toxicity, and for neuroblastomes, these 2 concentrations and these from orange were removed. And then what we did here was to run a simulation of the formation of the beta amyloid plaque, okay? And to test the 5 strats at the concentrations that we know are not non-toxic. This is just to show you how the neuroblastomes, when are together with the alpha beta amyloid peptide, reduce the cell viability. But using the strat, this viability, as you can see, is recovered. In the case of olive leaves, was very good, the recovery. So that's why we select this for the next lipidomics study. And to do this, we just use this approach to extract the lipids with methanol methyl terbutyl ether and this LC with this column, this CSIC. And as you can see, we were able to find 253 lipids here, combining the mode of interface positive and negative of the electrosprite. And we were able to differentiate here the neuroblastomes control and the treated with the strats. And as you can see, many of these were related to these compounds. I never remember the names. Phosphatidylcholines, phosphatidylethanolamines, okay? And many of these triglycerides decrease here. So this is just the chemical similarity enrichment analysis to show you this is decreasing, some of them were increasing a lot. And what was important is that it was correlating very well with those cases found in the literature in which these were related to a better behavior of Alzheimer people, okay? And next, what we did was, once we have observed these strats have a good activity, we confirmed this activity using this transgenic C. elegans, this CL4176, which is a model of Alzheimer, okay? And what we could see were very nice results here. And I mean, this was done by a company who has this transgenic C. elegans. We do not have it at the lab. And they were really impressed because the positive sample that they use for checking this model is this from ginkgo biloba at 100 micrograms per milliliter. And as you can see, our strats from orange or from Dunaliella salina at 50 micrograms, so the half of the concentration, gave better results in terms of the recovery of the mobility of these C. elegans. So what we did then was to investigate how was this part done. We did this by transcriptomics. We did also by metabolomics, combining the lipid profiling and the primary metabolism profiling. So this was done in this case. In transcriptomic, we found 150 genes significantly altered, okay, using a false discovery ratio of 0.05 and a false change of 1.5. 120 were upregulated, 30 downregulated. These are the main biological processes involved, okay? In this worm base, we could find the different genes just linked to these biological processes. And this is the pathway enrichment analysis that shows these processes which were mostly altered by the use of the strat in the C. elegans. And in this case, we ran the lipid and primary metabolomic profiling. This is the number of lipids that we found altered. Sorry, we found 524 lipids and 195 metabolites were identified there. And finally, 63 were significantly altered by using these parameters. So these are the intracellular primary metabolism and intracellular lipid profiling. This is just the integration of the transcriptomics and metabolomics analysis that go to indicate this as the main biological process that were modified. And of course, what we are now running is the proteomic analysis of the C. elegans that we have enough amount just to extract the proteins and to do it. And I hope I will tell you this next meeting. And this is just the general conclusion, okay? I hope I have shown you that this was a good example of the important challenges that still have to be addressed in the future to take advantage of the full potential of the binomial food and health. And just to thank the group, this is the last barbecue, and thank all of you for your kind attention.